Hello there, and welcome back to Science for Sleep. Tonight, we are going to explore something that is happening to you right now. Not tomorrow, not when you eventually fall asleep. Right now, as you settle in and let the day begin to release its hold on you. Because the process we are going to explore tonight does not wait until you are fully asleep to begin. It starts the moment you begin to slow down. The moment you allow yourself to be still. Your brain is preparing to rewrite itself. Every single night, while you are completely unaware, while you have no sense of effort or intention, your brain performs an act of extraordinary biological engineering. It evaluates everything that happened to you during the day. It decides what to keep and what to release. It strengthens the connections that matter and quietly dismantles the ones that no longer serve you. It processes emotions that would have overwhelmed you if you had tried to face them while awake. And it does all of this through mechanisms so elegant, so precisely choreographed, that neuroscientists who have spent their entire careers studying sleep still find themselves moved by the complexity of what happens every night inside the sleeping human brain. The word neuroplasticity describes the brain's capacity to change, to reorganize, to form new connections and prune old ones, to be shaped by experience. For most of the 20th century, scientists believed that this capacity was largely limited to childhood, that the adult brain was essentially fixed, its wiring set, its potential determined. We now know this to be wrong. The adult brain retains an extraordinary capacity for change throughout life, and the primary mechanism through which that change is consolidated, organized, and made permanent is sleep. Tonight, where are you listening from? Is it late where you are? Is the room settling into quiet around you? Then your brain is already beginning its work. So find a comfortable position. Let your breathing slow. Let your muscles release their daytime tension. And let's explore together the most extraordinary renovation project in nature. The one that happens inside your skull every single night while you sleep. Let's begin. The word plastic in the context of neuroscience does not mean artificial or cheap. It comes from the Greek word plastikos, meaning capable of being shaped or molded. Neuroplasticity is the brain's inherent capacity to reorganize its own structure and function in response to experience, learning, injury, and the passage of time. The adult human brain contains approximately 86 billion neurons. Each neuron can form thousands of connections with other neurons through junctions called synapses. The total number of synaptic connections in a single human brain is estimated to be in the hundreds of trillions. This network is not static. It is a living, dynamic system that changes continuously in response to what you experience, what you learn, what you feel, and crucially, how you sleep. For most of the 20th century, the dominant view in neuroscience was that the brain's wiring was largely fixed by early adulthood. But experiments that began in the 1960s and gathered momentum over the following decades dismantled this view. Researchers studying patients recovering from strokes discovered that the brain could recruit entirely new regions to take over functions that had been lost when specific areas were damaged. People who had lost the ability to speak following strokes sometimes recovered language function through the recruitment of neighboring brain regions that had not previously been involved in language. The brain had rewired itself. Studies of musicians found that the cortical regions dedicated to the fingers used in playing their instruments were measurably larger than in non-musicians. London taxi drivers who memorize an extraordinarily complex urban geography were found to have measurably larger hippocampi than non-taxi drivers. The adult brain retains significant plasticity throughout life, but the question remained, when does this reshaping occur? During the active, effortful hours of experience, or somewhere else? The answer is sleep. In 2003, two neuroscientists, Giulio Tononi at the University of Wisconsin and Chiara Chiarelli, proposed a theory about sleep that has become one of the most influential frameworks in sleep science. They called it the synaptic homeostasis hypothesis. The hypothesis proposes that wakefulness is fundamentally a period of synaptic strengthening. During the day, as you experience the world, learn new information, and respond emotionally to events, the synapses in your brain are being strengthened. This strengthening is the molecular substrate of learning. Synapses that are repeatedly activated become more efficient at transmitting signals. But synaptic strengthening requires resources. It costs energy. It takes up space. If every synapse activated during a day of waking experience were simply made stronger without any counterbalancing process, the system would eventually saturate. The brain's ability to discriminate between important and unimportant information would collapse. Tononi and Chiarelli proposed that sleep is the brain's solution to this problem. During sleep, and particularly during deep, slow-wave sleep, the brain performs a systematic downscaling of synaptic strength throughout the cortex. Not randomly, not uniformly, but selectively. The synapses most heavily activated during the preceding day, the ones most connected to important experiences and new learning, were relatively protected. The synapses only weakly activated, the background noise of daily experience, were pruned back. The overall level of synaptic excitability was reset toward a sustainable baseline. This downscaling restores the brain's capacity for new learning by preventing the saturation that would otherwise accumulate, and it actually improves the relative strength of the most important connections. When the background is reduced and the signal remains, the signal becomes proportionally clearer. The memories of the most significant experiences of the day are not simply preserved, but highlighted, their relative prominence increased by the reduction of surrounding noise. This is what the slow waves of deep sleep are doing. They are the mechanism of a nightly recalibration, a planetary scale editing process conducted across hundreds of billions of synaptic connections, reshaping the brain's connectivity landscape in ways that make the next day's learning possible. Memory is not a recording. This is one of the most important and least appreciated facts about how the brain works. When you experience something during the day, it is encoded as a pattern of synaptic activation. The hippocampus plays a central role in this initial encoding, binding together the disparate elements of an experience into a coherent whole that can later be recalled. But the hippocampus is not designed for long-term storage. It is a temporary repository where new memories are held in a fragile, recently formed state. Over time, with sleep, memories are transferred from the hippocampus to the neocortex, the vast outer sheet of the brain, where they are stored in a more distributed and durable form. This process is called systems consolidation, and it is critically dependent on sleep. During deep sleep, the hippocampus replays the experiences of the day in a compressed, accelerated form. Sharp wave ripples, brief bursts of high-frequency activity in the hippocampus, occur hundreds of times each night. Each one carries fragments of the day's experiences upward into the cortex, where they are integrated with existing knowledge. This integration is not passive. The cortex actively makes sense of what the hippocampus sends. New information is connected to related memories already stored in the cortical network. The meaning of an experience, its connections to what the person already knows and cares about, is worked out during sleep in ways that cannot happen during the busy hours of waking life. This is why sleeping on a problem so often produces insight the following morning. The brain has not simply rested, it has been working, quietly connecting the problem to relevant knowledge and experiences that the waking mind had failed to access. A remarkable series of experiments conducted by Jan Born and colleagues at the University of Lübeck demonstrated this directly. Subjects were trained on a mathematical problem-solving task that had a hidden shortcut solution. None of them discovered the shortcut during the initial training session. After a period of either sleep or equivalent wakefulness, they were tested again. Subjects who slept were approximately 3 times more likely to have discovered the hidden shortcut than those who stayed awake. The sleeping brain, processing the problem during the night, had found the elegant solution that the waking mind had missed. If is neuroplasticity in action? The brain encountering a problem, sleeping, and waking with a restructured understanding of the problem's solution. The rewiring that happened overnight was directed, purposeful, and extraordinarily effective. Not all of what the brain processes during sleep is cognitive. Much of it is emotional. And the emotional processing that happens during sleep may be among the most important contributions of sleep to psychological health. Matthew Walker has described the role of REM sleep in emotional processing as overnight therapy. During REM sleep, the brain's electrical activity resembles waking activity so closely that an EEG recording is difficult to distinguish from one of a waking person. The dreaming brain is intensely active, processing memories, generating emotional experiences, and rehearsing social scenarios. But there is one crucial difference. During REM sleep, norepinephrine, the brain's primary stress neurotransmitter, is almost entirely absent. The locus ceruleus, the brain region responsible for releasing norepinephrine during states of alertness and arousal, essentially ceases its activity during REM sleep. This is the only period in the entire 24-hour cycle when norepinephrine drops to essentially zero. What this means is that during REM sleep, the brain reactivates emotional memories in a neurochemical environment stripped of the stress chemistry that made those memories distressing. The memory is there. The emotion associated with it is accessible. But the raw physiological alarm response that accompanied the original experience is muted. This combination, emotional memory reactivation without stress neurochemistry, is what allows the sleeping brain to process difficult emotional experiences and reduce their capacity to harm. The memory is revisited, its emotional content is worked through, but the person wakes the next morning with the memory intact and its painful edge softened. This is why difficult experiences feel less overwhelming in the morning than they did the night before. This is not simply the result of time having passed. The brain has literally changed its response to the memory. The synaptic connections that encode the emotional response have been recalibrated. The rewiring is real. When this process fails, the consequences are severe. Post-traumatic stress disorder involves, at its neurological core, a failure of this nightly emotional processing. The traumatic memories are reactivated during sleep, but instead of being processed in the quiet, norepinephrine-free environment of healthy REM sleep, they are experienced with their full emotional intensity intact. People who are deliberately deprived of REM sleep show dramatically increased emotional reactivity the following day. Their amygdalas become more than 60% more reactive to emotionally charged stimuli. The prefrontal cortex shows reduced connectivity with the amygdala. The emotional brain and the rational brain become less able to communicate. The rewiring that REM sleep performs is not a luxury, it is a daily necessity. The brain's capacity to consolidate and improve learned skills during sleep extends far beyond explicit memory. Some of the most compelling evidence for sleep-dependent neuroplasticity comes from studies of motor learning. When you learn a new physical skill, the initial learning is effortful and imprecise. You are consciously attending to every component of the movement, correcting errors, gradually building a representation of the skill in your motor cortex. After sleep, something changes. not just in the sense that you are rested. The neural representation of the skill has been reorganized. Studies using neuroimaging found that after a night of sleep following motor skill training, brain activation during performance shifts. Regions associated with effortful, conscious control of movement show reduced activation. Regions associated with automatic, fluent execution show increased activation. The skill has been transferred from one neural system to another during sleep. This transfer reflects a fundamental change in how the brain is executing the skill. What was previously effortful and consciously controlled has become more automatic, more efficiently encoded, more accurately executed. The skill has been moved from the brain's working memory to its procedural memory, from the slow and effortful to the fast and fluent. Sleep spindles, those brief bursts of activity in the 12 to 15 Hz range that occur hundreds of times each night, have been specifically linked to motor skill consolidation. People who show more sleep spindles after motor learning show greater overnight improvement in motor performance. And the spindles are particularly concentrated over the motor cortical regions that were activated during the preceding day's learning. The brain is targeting its consolidation processes precisely where they are needed. One of the most counterintuitive discoveries in neuroscience is that the brain grows by eliminating connections rather than adding them. During development, the brain produces an enormous excess of synaptic connections, far more than the adult brain will retain. This overproduction is followed by a prolonged period of synaptic pruning, in which connections that are not being used are systematically eliminated. The brain sculpts itself through loss. This pruning process continues in a more moderate form throughout life, and it happens predominantly during sleep. The process is governed in part by a class of immune cells in the brain called microglia. During sleep, microglial cells actively survey the brain's synaptic landscape, identifying synapses that are weakly active or structurally compromised and eliminating them. A landmark 2017 study found that microglial pruning activity was dramatically increased during sleep compared to wakefulness. If the brain's pruning machinery is most active during sleep, then chronic sleep deprivation may impair this process, allowing unnecessary or damaged connections to persist when they would otherwise have been eliminated. Studies in animal models found that prolonged sleep restriction leads to excessive synaptic accumulation and measurable cognitive impairment. The pruning did not happen. The connections that should have been removed remained, and the brain's performance declined accordingly. The brain you wake with each morning is literally a different brain from the one you fell asleep with. In the precise configuration of its hundreds of trillions of synaptic connections, it has been edited, refined, pruned of what was unnecessary, and consolidated in what mattered. The culture of overnight study, of pulling all-nighters before exams, rests on an implicit assumption that more time spent reviewing information will result in better retention. The neuroscience of sleep consolidation suggests that this assumption is not merely incorrect, but precisely backward. Learning requires two stages: acquisition, during which new information enters the brain and is initially encoded, and consolidation, during which that information is transferred to long-term storage and integrated with existing knowledge. Acquisition can happen while you are awake. Consolidation, in its most powerful form, requires sleep. When you stay awake to study instead of sleeping, you are gaining marginal additional acquisition time while sacrificing the consolidation process that would have turned that day's learning into durable memory. Robert Stickgold at Harvard Medical School found that a 90-minute afternoon nap containing both non-REM and REM sleep could produce learning consolidation equivalent to a full night of sleep. And sleep not only consolidates what has been learned, but actively protects it against subsequent interference. Memories consolidated during sleep are more resistant to being overwritten by later learning. Sleep does not merely preserve memories, it stabilizes them. There is a network of brain regions that activates when the brain is not focused on any particular external task. When you let your mind wander, when you daydream, when you think about the future or reflect on the past. This network, called the default mode network, is involved in some of the most sophisticated cognitive processes the brain performs. Self-reflection, mental time travel, theory of mind, the construction of narrative identity. During REM sleep, the default mode network shows patterns of activity that resemble its activity during waking mind-wandering. The sleeping brain is not simply replaying the day's experiences like a recording. It is interpreting them, constructing narratives around them, connecting them to the self's broader story, working out what they mean. This interpretive activity is part of why dreams feel narrative rather than random. The dreaming brain is constructing experiences, drawing on the day's memories and the brain's broader emotional landscape, and fashioning them into something that has the structure of a story. The rewiring that happens during this process is the rewiring of self-understanding. The brain does not merely consolidate factual memories and motor skills during sleep. It also works, night after night, on the more complex project of integrating new experiences into a coherent sense of who you are and what your life means. Sleep is not only a biological process, it is, in part, a narrative process. And the story it is editing is you. The brain's capacity to rewire itself during sleep is not independent of how you live. The conditions you create for sleep profoundly influence the quality and completeness of the neuroplastic processes that occur within it. Consistency is perhaps the most fundamental condition. When sleep timing varies by more than an hour or two from night to night, the circadian regulation of sleep staging is disrupted. The proportions of deep sleep and REM sleep shift. The nightly sequence of neuroplastic processes is compressed or altered. Temperature has a direct and potent effect on the brain's ability to enter and sustain the slow wave sleep that underlies synaptic downscaling. A cooler sleeping environment supports the core temperature decline that initiates deep sleep. Stress management in the evening hours is particularly important for the emotional processing that happens during REM sleep. High cortisol levels at bedtime fragment sleep architecture, disproportionately reducing the REM sleep that performs overnight emotional therapy. Physical exercise consistently increases the depth and quality of slow-wave sleep on subsequent nights. More powerful delta waves, more complete synaptic downscaling, and more effective memory consolidation. And perhaps most important of all, simply protecting the length of sleep. The brain's nightly renovation has a timeline. It needs the early night for deep sleep and synaptic downscaling. It needs the late night for the extended REM periods that perform emotional processing and creative integration. Truncate either end, and the work goes undone. The brain you aspire to have, the one that thinks more clearly, remembers more accurately, feels more stable, and responds more creatively, is not created through more waking effort alone. It is created in equal measure by what happens during the hours you cannot observe. The hours when the lights are out, the body is still, and the most sophisticated engineering project in the known universe quietly proceeds without your knowledge or participation. Those were the sleep science facts about rewiring your brain while you sleep. And now, something is changing in the room around you. The words are coming more slowly. The thoughts that were following them are beginning to drift. There is a quality to this moment that is neither fully awake nor fully asleep. That threshold state that the brain inhabits for a few extraordinary minutes each night as it crosses from one world into the other. Your hippocampus is already beginning to index the day, the events, the conversations, the moments of frustration and warmth and surprise that accumulated between this morning and this moment. The slow oscillations are preparing to begin,
those long, sweeping waves that will wash through your cortex in the hours ahead and carry the noise of the day down to a level where the signal can finally be heard. The microglial cells are readying themselves for their nightly survey, moving through the intricate landscape of your synaptic connections, attending to what is strong and what is weak, what to preserve and what to release. The norepinephrine is beginning to withdraw, and with it, the sharp edge of whatever the day left behind. The amygdala will have its say during the night, but in a quieter voice, in a biochemical environment designed not for alarm, but for resolution.
The default mode network is beginning to stir, preparing to do what it does in the hours of dreaming, constructing the narratives that make sense of experience, connecting the new to the old, weaving the threads of today into the longer story that has been accumulating since you were born. You do not have to do any of this. It does not require your effort or your attention or your understanding. It requires only one thing from you: the permission to sleep. The willingness to be still for long enough that the ancient, intelligent processes of the sleeping brain can do what they have been doing for every night of your life and every night of the lives of every human being who ever existed, and every night of the lives of the animals we descended from across hundreds of millions of years of evolution. Your brain is not finished with today yet. It has work to do with it. Important work. Work that will make tomorrow's thinking clearer, tomorrow's emotions more stable, tomorrow's learning more durable, tomorrow's understanding of yourself a little deeper and a little more integrated than today's. Let it work. You only have to let go. Let your breathing find its own pace. Let the slow waves come. Science for Sleep will be here when you return.